Next speaker is Jesus Vilar, again, and he'll be talking, I think, passionately again, but about genetics this time.
So I was invited to talk for around an hour and a half, no? According to, no, okay. In the next 30 minutes or so, I'm going to talk about, this morning some of the speakers already introduced some comments and terms on genetics. When you talk about molecular biology, you are really talking about genetics as well. So in order to facilitate the comprehension of this talk to those who really has no idea about genetics or who are really panic about genetics or who don't like to talk about genetics, so just to tell you a few introductory definitions and knowledge. For example, we know already that the human genome is composed by 3 times 10 to the 9, because I had to say that in terms of mathematics, because for the American it's 3 billion, for European it's 3,000 million nucleotides. Okay, and 0.5% of those 3 times 10 to the 9 nucleotides differs among individuals. And in fact, that difference, that 0.5% only, is responsible for all the phenotypic difference among us. That goes from, you know, the hair, the color of the hair, to our weight on height, you know, so I am short, so I assuming that is in my genes to be short. And from the even response to treatment or response to drugs, or even the way you manifestate any disease. So really, this is only the response, 0.5% of that nucleotides. So you should note as well that phenotype, the phenotype difference among us is a consequence, is a direct consequence of a genotype, of the genotype variance or genotype differences. So it's really, our destiny is really in our genes. I say that because that will help us to understand this case. This is a real case. This is a patient. Well, in fact, this is 2 patients. They are treated in the same ICU by the same physicians. They have the same problem. They were admitted because they have a severe pneumonia caused by the same bacteria. And they are almost the same age, both the same gender. Were treated, I said, the same way, in a very few months apart. And those both develop sepsis. They require vasoactive drugs, you know, and they develop ARDS, as you see in the X-ray. One of them survived, and the other one died. So when you take that in consideration and you want to see what happened in your ICU and you collect information from patients with or without pneumonia, in this case with pneumonia, who develop severe sepsis, you say, okay, I have, you know, last year I have 100 patients, our mortality rate is 50%. And this is how you present the data into meetings. However, this is really, I would say, I feel very uncomfortable. And then every year, I feel more uncomfortable about this way of presentation, the data. Because we will see later on that we have to find an explanation why the other patient died, being cared by the same physician and by giving the same treatment. The patient had the same severity degrees. So, you know, it's not a question of lack of chance. We have to explain the lack and the chances. And I think we are into that issue with the technology that we have today. Because until recently, the way to deal with diseases, the way to understand, the phenotype, that is how the disease is presented, how it's going to be solved, how it's going to progress, is always have been thought to be only dependent on 2 factors. You know, the environment, and the clinical presentation of this, based on the way we measure today that symptoms, the duration of disease. This is how we write that in the clinical record, how respond to treatment. But more and more evidence support the fact that the phenotype is a direct consequence of the genotype. And in fact, not me, because I am not an authority in this field, but it's been said that 60% or more of the disease risk or disease presentation is caused by the environment plus the your genetic background, what you have already printed in your genome. This is as well represented in this graph. What you see here, the environment and the human behavior. I mean by that, if you are a smoker, you are alcohol drinker, whatever, but it's with your genes will really determine the disease presentation, progression, remission, whatever. In fact, in many complex diseases like the pneumonia or sepsis, that are the most common causes of ARDS, what happens is the genetic variants, first of all, they are due to multiple genetic variants. And that multiple genetic variants confer an incremental, a cumulative risk to have a disease, to develop that disease. This is why now, and it's going to be very near in the near future, the medical students and the physician are going to study not only the organs, not only the macroscopic or microscopic, anatomy, but they are going to study gene anatomy. We are going to explain things not all by organ dysfunction that I think already Dr. Slasky talked this morning, and now, with lung injury as a cause of any other organ dysfunction. But we have to explain things more in detail by talking about gene dysfunction, because I think in the future therapy will come in that direction. Genes that now many of us, we don't have this knowledge because we are not trained to that, but it's, you see clinical journals today. Clinical journal is very rare that in clinical journals, medical journals, you don't find a paper, a clinical research paper dealing with genes. So this is really, now it's very unusual. More and more clinical journals publish genetics or research dealing with genetics in patients. So genes, so the only thing that I would like to remember from you in my talk today is the genes. Imagine this is the chromosome, the chromosome 8. So a part of the gene has a short and a long arm. In this case, because we are talking about the C-myc oncogene, but forget about the name, it's just the way that you are going to see represented the genes will be like a line, and you see the boxes here that represent the exons. Exons mean the part of the gene that codes part of the protein, the entire protein, because the gene is responsible for to produce a protein, usually with the same name, and so every exon is responsible to produce a part of the gene. When at the end of this process, all this information is combined and produce the entire protein. But in between the exons, you find spaces in mammals, but not in bacteria, that we call introns, that is supposed to be garbage DNA that without any function, and now we know that if there is for some reason, it's because we didn't know in the past, but now we know more about this. So you have a gene, you have exons, here you have 1, 2, 3 exons, and you have introns, and you have numbers here. But the most important area that I like to focus is before the gene starts, because a gene starts with the exon 1, of course. But before the gene is a region that you are going to see in many papers. It's called promoter region, okay? This is a region that has not any, it doesn't code for any part of the protein, but it's important to tell somehow to activate the gene and say, hey, go, and do this. And in that case, to do this means be activated and produce the protein, or activate it and go to some other place. We don't know too much about, or even we know very little about how genes work because they respond so fast. Even, I don't know if faster than the speed of light, but my God, they respond so fast that we don't know how they do it in the complex 3 billion or 3,000 million nucleotides. So remember, the promoter region, the exons, and the introns. Now, if I said before that in the gene is our destiny, I have to prove somehow, in some cases, how that is really happening. For example, the first paper dealing with this idea that diseases is a consequence of genetic variation was this paper published in 1988, when still technology to sequence the gene were not developed. And this is a paper that has nothing to do with genetics. In sense, it's just an epidemiological study. They was done in Denmark with almost 1,000 families. They have a great registry of almost everything. And this is a registry from 1905, last century, in which they went to the registry by the government and about 1,000 families who adopt a child. What happened is they knew from 1905 who are the biological parents and who are the adoptive parents and who are the child, a boy or a girl. And they know already, they knew how many of them are alive, how many of them are dead. Not only the adoptive, but the biological parents as well. They have that information. So they ask for permission, and they follow very simple. They want to know those parents and adoptees who have been dead by that time, by the late 80s, a relation between, to be an adopted parents to a biological parent with the death of the adoptees. So this is what they did. So they classified adopted parents, biological parents, and kids in 4 groups of diseases. One was cancer, the other one was cardiovascular diseases, the other one was infections, and the other one was miscellaneous. And when they got all the data together and they tried to analyze the risk of death, they found something that they didn't expect. In fact, they were expecting that cancer and cardiovascular diseases were the most related genetically diseases in terms of between the adoptees and biological parent. But they found the number one problem was infection. In fact, they found that if biological parent died from infection before the age of 50 and 70, this is the 2 group of ages that they study, the mortality for adoptees increased 6-fold. So it was really a lot. That is something that's expressed in this figure, and the only time that they mentioned the word genetics was at the end. They said, premature death. Premature death because to die before the age of 50 is, I will call premature death. In adult has a strong genetic background. It's no other way to explain it because those people were living in a different environment with different parents. They have different way of life and different meals. I mean, everything was different. Education, everything was different. But however, besides those differences, biology has something to say. In fact, this figure reminds me when I think I first gave the talk in Spain, a pediatrician told me, this is how I really think, because pediatricians think that all the children they see are their children. The pediatricians say, my children. We don't say too many times my patients, but pediatricians say my children. The pediatrician said, my children, my 10% of the children has 90% of the diseases, in this case of infections. Because this is what the pediatrician really, this is what their practice are. I mean, they receive the same patient, the same kids over and over. And this is a group of kids they know very well because they have all the infection. So, and I'm talking about an infection because I put it in the example of the pneumonia. And because infection is the major causing, the major clinical condition associated with the development of ARDS, or kidney injury and ARDS. So if the genes, our destiny is in our genes, we have to find the variation in our genomes. And the variation today can be explored, analyzed, examined, and even known because we have the technology. Today, it's an amazing that the way we can deal with just by checking the number and the nucleotides, the position of the nucleotide. We can see that. Eventually, in the very near future, you are going to a shopping mall, a shopping center, with a drop of blood, even less than, even with the saliva, they can tell you, while you buy your pair of shoes or you do your shopping, you can take back your genome and tell the risk of, depending on the science that is published, the risk for any diseases that is associated with those variants. So this is, so the simple variant that you can get, and this is the only I'm going to talk, is it's called the SNPs, single nucleotide polymorphism. This is very weird word, but it's very simple. It's amazing. Just the change of one nucleotide. You can't believe it that in 3 billion or 3,000 million nucleotides, just the change of one nucleotide for another is going to make a big difference. I mean, if somebody tell me that a few years ago, I would say, forget it. I mean, a mutation, I understand. You have a mutation in the protein, and the mutation produce a protein that doesn't function properly, so you cannot get, there are proteins that are very basic for life, so I understand that. But in the entire genome, to have just a change of the letter is going to have a tremendous impact on diseases? Well, believe it or not, the answer is yes. And in fact, it's so important that now many companies are taking this information into the benefit to take better care with patients, because if we prove that some of the SNPs can be considered genetic markers, we talked this morning about biomarkers. Everybody wants to have a biomarker to be able to classify, diagnose, to better treat patients. If we can get some of those SNPs as a good markers of disease progression, diagnosis, remission, whatever you call, as predictors for clinical outcome, my God, will be a great business, a great business. And this is why many people are investing in that business. In fact, today we have an information about how the cells during sepsis is working. Sepsis, that is a major cause of ALI. We finally know the whole cascade, the signaling cascade. That is really, I don't go into that. But you will see that there are so many genes with many initials, forget about the names. But those genes are very well known today. They know already that all of these genes are polymorphic. Polymorphic means that they have gene variants. They have changes in those letters, and those changes are in the promoter regions, those changes are in the exons, and those changes are in the introns. So in many parts of several, in fact, in some of those genes, in many parts of that gene. Now, for example, the LBP, TLR4 and cytokines, this is the one I'm going to talk today. Do you remember the patient that I mentioned to you before? Well, it turns out that we know now that the TNF is a very polymorphic gene, like any other genes that I put in that graph. Well, what happened is that the TNF, the normal TNF gene, in the position minus 308, minus mean that is in the promoter region, is before the gene starts. In that position, you clone the gene, you sequence the gene, and instead of having a G, some of us here in this room have an A. And this A give you, you produce a different kind of gene, a different kind of protein, that just to put a different name, they call it TNF2. So normally, the wild type is TNF1, and now you have the TNF2. So, okay, let's go to that patient. So that patient, we keep some blood from that patient, from those two patients, and when we analyze the genome for that particular gene, for the TNF, and we sequence the TNF in those patients, we end up knowing that this person here has the TNF1, the one that most people have. But this one, the second patient here, have a TNF2. And what is the difference between these TNF2? Well, this patient died, remember? What happened is individuals, you don't need even to be a patient. People who has the TNF2 by nature produce a tremendous amount of TNF when they activate it. So what happened is, really, when you have sepsis, and you have the bad luck, I would say, to be the TNF2 person, you live in a cytokine storm. So really, your body is producing tons of TNF. Now you can see why patients with ARDS due to sepsis in the past, when they receive anti-TNF, the study was negative. It was negative because they never took in consideration that is two kinds of patients. In these patients, if you block TNF, maybe you can kill the patients because TNF is important. But in these patients, those patients are the ones in which should block this amount of TNF. That happened, like I said this morning, you know, this is, I think, why our study, I think, is important to the clinicians. We are able to identify risk patients at 24 hours just by putting the patient on a standard ventilatory setting. Now I think in the near future a company is going to develop a drug to fight this cytokine storm, are going to ask the physician, please tell me which one of these two patients is the one I'm going to give the drug. Because according to the clinical data that I have no way to differentiate these two patients because they have the same Apache, they have the same disease, the same culture, everything is the same. Maybe you measure the cytokine level are very high, but it's an it's an overlapping between cytokines level because in every patient with sepsis, they have high levels of TNF. Do I have any proof that this is the case? Well, yes, this is a paper published a couple of years ago by the group in Boston, when they analyzed now 212 patients with ARDS. And what they saw is the following. By just measuring what I said, just in the minus 308 position of the TNF gene, if you are homozygous for G, that is, I think, what normal people in this room is, or you are heterozygous or homozygous for A, what happened is patients in this group have the highest mortality rate, close to 80%. So again, it's another way to deal with the mortality rate. Of course, the mortality rate in their series was 40-something%. But again, doesn't tell you anything. If I have an interest in invest in a drug, I would like to know what kind of patient you are. Because this patient, for example, this group of patients with a mortality rate 40% already, I have to do something else. Of course, nothing related to the TNF. I can improve survival here, like almost 40% survival. I have a tremendous gap of improving survival in this group of patients. I think tomorrow, maybe Arthur Slasky is going to talk about future directions to treat patients with ALI, ARDS, in the setting, in sepsis or no sepsis. Another cytokines, yes. I don't want to, to be into big details, but other group of people in Vancouver, for example, physician Shelton and co-workers in the Gene Russell group, they're looking for interleukin-6, but instead of looking for one SNP only, they sequence the entire gene and they look for all the SNPs that they have in the gene. And they group those genes in what is called haplotypes. Haplotypes are patterns of several genes that are inherited together. So it's much better, they give you more information. And what happened is, and the geneticists, you know, they can draw, I cannot explain that to you now because it's going to be more complicated. But what happened is they have a table, they can see, you know, all these SNPs here across the entire gene. And they, by the combination that they already have the information, they can classify in haplotypes, and it's called even some of them clades. So pay attention to this haplotype, the GCG. That means the GCG means that 3 SNPs are together and they inherit together. Okay, what happened is in this particular haplotype that is in the gray, are patients with the gray bar here. Those patients with the gray bar, they have homozygous for that haplotype. They have the more incidence, let me see, more incidence of ALI. This are not even septic patients. They are patients with SIRS. SIRS is almost every critical ill patient in the ICU. I don't think you can, I mean, I think all the patients in the ICU have SIRS by definition because, you know, to have a little bit of fever and leukocytosis, almost everyone has. But anyway, so this patient, 228, even you see the phenotype was very broad. So they have more incidence of ALI. They require more ventilation. They is on mechanical ventilation. They require more vasopressors, and they have a more severe cardiovascular dysfunction. It's amazing how just by having that genotype. There's no other way to explain the difference. Other proteins, other genes, yes, LBP. LBP is important because it's the gene that is where it's the protein that binds to the endotoxin. And the only study so far that is published is this in the 2002, where having a polymorphism in that position in the amino acid, in the codon 98, by changing one of the amino acid, you know, the susceptibility to sepsis in male is higher. But now we are preparing this manuscript because we clone and sequence the entire LBP gene, and we have identified some published, but there are other unreported SNPs in that gene, and we found that it's a couple of SNPs that they inherit together, so really is an haplotype, that give you like a almost 8, sorry, no, not here. It's like, it give you like a 5, the risk of developing ARDS is like a septic ARDS is like a 4 times more than if you don't have that haplotype. Toll-like receptor, that is the specific receptor for sepsis, but I'm going to pass. But another, this morning, I think Rolf Humeyer presented in a slide in when he mentioned the nuclear factor kappa B, and he mentioned, I think, one of the chemokines. While the chemokines are important, this is the CXCL2. You don't identify by this name the cytokine, but this cytokine was known before as a MIP2, the macrophage inflammatory protein. But you know, the genes, according to regulations, they change names. So this is an important chemokine because it has a potent chemoattractive properties and has many effects on inflammatory and immunologic cells, especially neutrophils. What happened is we cloned this gene for the first time. We sequenced the entire gene. We identified several SNPs, and we found out that one in the position -665, mean in the promoter region of the gene. We here, people has different numbers of what is called a tandem sequence repeat, mean that in that position, people has 1, 2, 3, 4, or even 30-something copies of the AC, many ACs. People don't know why that is important, but in that position, maybe I have 2 copies, you have 5 copies, Marini have 24 copies, that is good if Marini have 24 copies, because we end up knowing that if you have 24 plus minus 1 copy, even though you have the risk to develop septic ARDS is higher, it gives you higher, is like almost 4 times higher, they give you a protective effect. A protective effect. And this is something that has come through the evolution. It's funny. So you have now an SNP that multiply the risk of having a disease for 3, 4, even 7 or 10 times. But that risk is associated now with a much better progression of your disease, much better evolution, response to drug, response to treatment, so, and cure. So in that case, patients who were homozygous for this 24 plus minus 1 had the lowest mortality rate. And I'm talking about severe sepsis, ARDS associated with severe sepsis. Compare with those who don't have that 24 copies. You can have 6 copies, 11 copies. I don't care how many copies do you have. But if you don't have the 24 plus minus 1, the mortality rate, without taking in consideration other factors, is more than 70%. So it really is a big difference because it's now more than double. Sorry, not more than double, almost double. So you see, just by looking at the genetic background. Well, I will escape this. Limitation of the studies that you are going to see published in the journal. There are some limitations. One of the limitations is important because it has to do with the ethnic background. And a study that could be positive in Europeans could be negative in Asians. In fact, a study could be positive in North Europe, can be negative in the south of Europe. And that, for example, is the case of an example I'm going to show you later. So because we don't have the same genetic background, through the evolution, through isolation of populations, we develop our own, not only we develop, we have maybe the same variation, the same SNPs, but the frequency of the genotypes are not the same. So really this is why some studies are limited by the number of patients who are well stratified by having all the genotypes. The other is that, like I show you in the MIP2 and the CXCL2, is that the genes who give you, who multiply the risk for susceptibility, for predisposition, are not the same genes that are involved in disease progression. Not only that, the other thing is that maybe it's not the SNP that you are looking at, the one that is responsible for the association. It's other genes who are linked together, they inherit together, but because you don't know which one it is, but very soon you will know because systems biology is a field that is developing and they want to see the network of all the SNPs. They will tell you more information. The other thing is, maybe you study patients with sepsis and you find negative results, but if you close down the phenotype to severe sepsis, now you can get positive results. So depend on the phenotype. Maybe if you study ALI patients, you don't get any result, but you study ARDS or establish ARDS, you get something. So really, the phenotype is very important. The other thing is sample size, because it's very difficult, and now it's becoming more and more difficult to have patients with ARDS. So really, most of the studies that have been published in the past lack power, and this is why it cannot be reproducible. This is the example. For example, this is the case of the angiotensin converting enzyme. This is an enzyme that converts angiotensin 2 to 1, and not only that, degrades bradykinin. And bradykinin is important for the vascular tone. It has been thought that because the lung is the largest endothelial surface in the body, that this gene is important in the development of ARDS, and was studied by a British group, Marshall, and published in the Blue Journal in 2002. And they study a very specific polymorphism. It's called insertion deletion. Insertion deletion means that it's a portion of the gene that can be or cannot be there. So it doesn't affect that you are healthy or unhealthy. It's just nobody knows. 289 base pairs, it's a big piece of the gene. Well, this is really a small piece, but for you will be a big piece because 289 is not like before I said AC is 2 only bases. Now it's 289. In the intron 16, remember I said in the intron mean the the portion of the gene people thought that was a garbage. Well, in the intron 16, to have that portion or not to have it, it gives you a different genotype. So patient can be homozygous for the insertion, they have it, or could be homozygous for deletion, or you can be heterozygous. You can have one allele with the insertion and the other without it. So what happened is they study 96 ARDS patients. I said this is a British population. And they compare the control group with ICU patients and general populations. What happened is they found that having the DD genotype, the deletion, by not having that part of the gene, the mortality rate was very high. Very high mean above 50%. This is patients who meet the criteria, the American and European consensus concept definition. However, look at this. Patients who have that insertion in both alleles, the mortality rate is very low, even lower than 20%, 12.5. During many years, that paper has been quoted because it was the only one published, until we published recently in intensive care the same study. We tried to replicate the study, but this time we used Spanish patients from Spain, and we stratify very well, and we calculate the genotype frequency. And we found out that we cannot replicate that study. In fact, the AC polymorphism has no any influence at all in ARDS patients with or without sepsis in our population, because, as you see for the sizes of the graph, there's not significant difference. So that is very important, as I said, not only to replicate the study, but as well in which population this study was done. So what is the future for that? That I don't think Arthur is going to have that slide in his talk tomorrow, but I'm going to give some insights. The future is that we don't need to be a patient, because the genetic information can be given to you just by giving a piece of, a small amount of DNA, because you don't need to be a patient to have that information. Your information is there since the moment you are born. And this is why I call unpatients. Genomic science, genomic medicine is going to change a lot the way medicine, or at least diseases, are interpreted. Because the nature of the disease had to be, I said, 60% or more of the disease is a consequence of the interaction between environment and genetics. So if you have a SNPs or an haplotypes or several haplotypes, because I think in this case ARDS, as being a complex disease, have multiple gene variants that confer, as I said before, confers a cumulative risk. So it's not a question that you need to measure only or to analyze your TNF. Maybe you have to analyze several genes. But anyway, whatever genes you analyze, it's very easy that some of those genes are going to be, different in you, and can explain a little bit the way, your disease is behaving. So by analyzing those SNPs, we are going to classify patients depending on those risks that are already been published. And by classifying patients in a different subgroups of patients depending on you are homozygous, heterozygous, for one or the other genotype, patients can be classified as members of a genetic tribes. Genetic tribes could be TNF, IL-6, LVP, CXL2, whatever gene, or even a collection or integration of several genes. That is really amazing. This is why companies around the globe, hospitals, very big groups that are interested in to integrate the clinical aspect with the genetic aspect. They are collecting samples, not only from the blood, but you can collect samples from any other part of the body to analyze and to compare. Maybe the comparison will be not so important comparing this with other patients with the same disease. And you can see what is the differential expression or the differential DNA. But Maybe you have to compare with the clinical picture with syndromes. So we are going to collect information as well for syndromes, because companies want to know why a patient with sepsis who do not respond to treatment, we can make it through the knowledge, the new knowledge that is going to develop or is already developing through the pharmacogenetics, a new field. We can make these patients to be a responder. This really is the whole issue of that. I'm not going to go into this, but I think many of you will agree with me that you would like to have this test before it's too late. So thank you very much. I have a genetics paper already, you see?
You better put it back up.
Eh?
You better be careful. It's only valid 2010.
Oof.
Thank you very much, Jesus. Any questions? Let me start with the first question. This is a small question. One of your very early slides, you had like a pie diagram, and you said the environment is 30%, genetics is 30%, and you said ethnicity was 1%. If ethnicity is not genetics and not environment, what is it? What is
Well,
it? What do you mean by ethnicity?
No, ethnicity means that there are some populations that by having the same variation, genetic variation, the frequency of that variation is different. So they have the same variation. But the problem is, for example, Japanese or Chinese, well, they have Chinese not because it's 1 million people, but the other, close population area in which those variants, some of those variants has been remain in the same population over and over, the frequency of the good or the bad genotype is very low.
Right, but it's still genetics then.
It's still genetics.
So,
it's still genetics.
You had it separate as genetics, you had environment.
Yeah, but I think it's related on the frequency, not really on other. This is why it's so small anyway. Eh? I put it as very
small because you don't know, huh? Yeah.
Maybe we don't know.
Rolf.
I was curious about your CXCL2 story.
Right.
And so you said if I have that, then my risk of developing a sepsis syndrome or an acute inflammatory response is much higher than the control.
Yeah,
but if I happen to have it,
yeah,
if I do get sick, I'm a healer.
You so get your survival, the risk to survive is higher.
Yeah.
Yes.
So that raises the question whether that's because I have other associated genes that heal me, or whether it's telling us that an exaggerated early pro-inflammatory immune response is actually a powerful repair trigger. Do you have any thoughts on this?
Well, say again the second part, Rolf.
So if you amplify
the first part, yes, I understand.
the early immune response, you get a lot of edema, you get sick, you get a lot of fever and stuff. But it also triggers genes that, at the same time, that really promote repair. And so this whole notion of separating inflammation bad and repair good, maybe it's...
Well, the issue here is, first of all, this is a case association studies, okay? And the limitation is you only study one gene, one SNP. Because in this case, we sequenced the entire gene, and we already analyzed the entire gene, so we didn't find that any other SNPs are associated with the one that I show you. That is a good thing, because I can tell you, there's no other SNP from that gene that is responsible for the effect. But it could be other genes that we don't know which one they are, that they are associated with, or they inherit with the same SNPs that we found in the promoter region that is really responsible for the effects. Now, about the CXCL2, what I can tell you that we know already is the animal studies that this is against the clinical results, but it could be in favor of what you said, because if you block CXCL2 activity, in animals, this is in animals, you improve survival in animals. This is really against what you find clinical, but maybe it's related to the time frame, as you mentioned. It's possible because people said, or investigators in the basic science said at that level on genetics, that CXCL2 is important for lung protection. I don't know in which way, but they said it's activated, highly activated during inflammatory process, and when you block it, you know, it's good. But in our case, we found in patients, by having that variation, in fact, the mortality rate was almost half.
Jesus, can I ask, well,
Again, it's a very, again, this is severe septic patients. So, it's an ARDS in severe septic patients. It's not, we don't include all septic patients, only the severe septic patients.
Jesus, two comments. One short. I noticed in your patient A and B, the chest X-rays look remarkably the same. They look remarkably the same for those two patients.
It's for educational purposes. The same.
When do you think, I mean, we've been hearing a lot about genetics. And so there's two questions. One is you talked about the fact that many studies that have been out there have not been able to be reproduced. You know, so you did what you showed one study was wrong. There's so many studies that are irreproducible.
Yeah, but
why is that?
There are not so many studies really in association. A case association study, for example, I tell you two of them. For example, the TNF has been reproducible in France and Germany. The IL-6 has been reproduced. The IL-10 as well. But then you will be amazed by very few really case association studies in ARDS. Very few in ALI. Very few. Compared to other diseases. Okay, like for example in rheumatology, there are many association studies, or even in cardiovascular diseases. But in ARDS, in ALI, I check very few studies, and all of them, you know, study the same, almost the same gene. You know, they insist on TNF, they insist on interleukin 6 or 10, and there are so many other genes to study. You know.
When are we going to, the last question, when are we going to be able to really use genetic studies in the ICU? I mean, right now it's an interesting theoretical possibility. When is it really going to be?
Well, I think we have to integrate that information. I don't think one gene can tell you everything. I think the system biology, to integrate 5 or 6 different genes that are involved in different cascades and network. Now we don't know. We think that they work separately, but they don't, because, you know, inflammation, coagulation, all these processes are at the same, simultaneously. We only analyze one aspect, one star over there, but that star is in combination with another here. So I think the future will come with all this is computerized, you know. Of course, we have now the chip. We can analyze thousands of genes at the same time, so we can see thousands of SNPs at the same time. Now it's expensive. But I think eventually this is what will come. The same way we analyze sodium, potassium, you know, in one drop of blood, very soon you will have the package of 1,000 genes that are involved in coagulation and inflammation, and you will see all the SNPs. And then, not only that, you keep that information, you store the information with the clinical syndrome that you have, and eventually somebody will be interested to compare, interact. Yesterday, Rolf was talking about syndromics. No, syndromics is another word, omics, you know, genomics, syndromics. Yeah, I think eventually will come. Very soon will come.
So the answer is eventually?
Eventually will come.
Oh, good. Okay, thank you very much.